All right, Destiny, it looks like it's just me and you. Do you have any questions?
No.
Okay, so the assignment, I don't know if you watched the previous lecture, but the assignment before was to do these crosses by doing Punnett squares. So I'm just going to do it out so that you guys can see this. But remember, when we do Punnett squares, each of the sides of the square just represent gametes that are produced by the parents. So if we look at this first one, what's the odds of getting a purple flower for the cross? Big P, big little p, big P, little p. So we know that the parents each have an allele for purple and allele for white. And that's from the previous thing that we did with Mendel. We know purple is dominant to white. So the rule is that you use the capital letter of the dominant trait to represent the dominant allele, and the lowercase of the dominant trait to represent the recessive allele. So this would be white, since it's recessive, even though it's a small p. And then this is purple.
It's not showing your screen.
All right, thank you. Okay, that should be better. So we have big P, little p, cross with big P, little p. Each of these parents have one purple allele and one white allele. And so they can only pass on one allele. So let's say this is the dad, it's going to make gametes. It can make big P gamete from this allele, on this chromosome, and then it could make a little p gamete, which would be white. So purple and white, essentially sperm, right? And then this would be the mom. So she could only make big P in one egg and a little p in another, and the ratio and the rate that would be made is 50-50. So there's a 50% chance this allele would be put in a gamete, and 50% this allele would be put in a gamete. And we learned all this in meiosis. All right. So the easy way to represent this is we just take all of the alleles that are available in each of the plants that they're going to pass down and package into gametes, and we just put it on Punnett square. So we can put big P here, and little p here, and big P here, and little p here. And so we can represent these as sperm and these as eggs. So if this sperm fertilizes this egg, we get big P, big P. This sperm fertilizes this egg, we get big P, little p. This sperm fertilizes this egg, we get big P, little p. This is a little p. And then this one would be little p, little p. So these are the offspring. Remember, each of this is a 50-50 chance, and we went over the rule of multiplication. So there's a 1/4 chance that this would be made. 1/4 this would be made. 1/4 this would be made, and 1/4 this would be made. And the rule of addition says we can add these together since they're the same. So that gives 1/4 plus 1/4, which would be 2/4. So we can look at, so what's the odds of getting a purple flower? Well, this is purple, right? It has both purple alleles, so it's going to be purple. So that's 1/4. Of them are going to be this purple. This is also purple. It has one purple and one white, but that's still, this is going to be dominant, so it's going to be purple. So we have 1/4 plus 1/4 from this, and then another 1/4 from this. And then this one, it doesn't count because it's white. These are going to be white. They don't have any purple alleles in them at all. And so 3/4 would be purple. And so that's the answer to the question. Odds of getting a purple flower from this cross is 3/4. And I'm just going to tell you guys right now that it's easier to know the ratios. So we know that this is what we call a monohybrid cross, and we've already done that. And so when we do a monohybrid cross, we know that the phenotypic ratio is always 3-to-1, 3/4 dominant and 1/4 recessive. And so I wouldn't even have to do a Punnett square with this. If I know the ratios, I automatically know 3/4 are going to be purple and 1/4 are going to be white from this monohybrid cross right here. What's the odds of getting the big P, little p genotype? Well, we can do this rule of addition here. We have 1/4 plus 1/4, which is 2/4, which is this reduces to 1/2. So the answer to this one is 1/2. What's the odds of getting the little p genotype? Well, there's only one way to get it, which is here, and that happens 1/4 of the time. So the answer to this is 1/4. But you could also, you wouldn't have to do a Punnett square on this either if you knew the ratios, because we talked about this. The genotypic ratio is 1 to 2 to 1. If you just memorize this ratio, you know 1/4 are going to have both dominant alleles, in this case big P, that 2/4 are going to have one dominant and one recessive, and that 1/4 are going to have both recessive. So I don't even have to do a Punnett square. If I know the ratio, I automatically know 1/4 are going to be little p, right? What is this type of cross called? It's called a monohybrid cross. All right, so this is a dihybrid cross because we're talking about two traits here. So I'm going to erase this. Okay, so we have two traits here, and we already did this. We did this cross back here. So this is the slide 16, where it does, we do the dihybrid cross, and it's using the same traits. Yellow is dominant to green, round is dominant to wrinkled. So same deal here. So big R is going to represent round, and little r is going to represent wrinkled. So we have this cross: big R, big Y, little y, round, wrinkled, yellow, green. And if you remember meiosis, these are, you can only pass on one allele for each trait, right? So like if your father had an allele for brown eyes and blue eyes, you would only get one of those. And if you had an allele for brown hair and blonde hair, you would only get one of those. And you couldn't get two alleles for your dad for hair color and none for eye color, because then your eyes wouldn't be right. So we have to pass on one of each, right? Remember, this is the gene for shape, and this is the gene for color. So we have to pass on one of each. Now, there's lots of combinations here because of independent assortment. Remember, it can line up different on the metaphase plate. Right here, remember, we could have moms on one side and dad on the other, and they could flip on this plate, so we can get all those possible combinations, which I kind of went over here on slide 16. So if you want to review that, you can. But I'm just going to tell you that let's say we pass on a big R, then we could also pass on a big Y, or we could pass on a little r and a big Y. And this is kind of like the math foil, right? So we're doing that inside and then the outside and then the inside and then the outside again. So we get little r, big Y, big R, little y, and little r, little y. So those are all the combinations of gametes. We assume these are not linked, so they're on different chromosomes. We don't have that linkage problem. And so you can do a Punnett square where each of these are the gametes that are made by, let's say this is dad. And this, let's say this is mom. And so you would have a much larger Punnett square. This would have 16 squares in it. And I'm not going to do them all out. If you want, you can go back and look at this, slide 16, and it has all of these gametes here and here, and the crosses that they make. This sperm fertilizes this egg, this is the offspring, and so on and so forth. So now we can address the question, what are the odds of getting a wrinkled green seed from this cross? And you could go through here and look. You could do the cross out, and you could look and you could say, okay, what's the odds of getting a green? I forgot the question. A wrinkled green seed. So we could go back and do the cross, and you see that this is the only way to get a wrinkled green seed. But if you know the ratios, again, if you know that this is a 9:3 to 3:1 ratio, and it's really in your best interest to memorize these ratios, you don't have to do this whole Punnett square, because you already know that this 9 represents both dominant. These 3 represent 1 dominant and 1 recessive in either order, right? So one could be like green, and that would be the recessive trait. So the other one had to be dominant. So that would be round. And then the other one would be yellow, which would be 1 dominant. So the other trait would have to be recessive, which would be wrinkled. And then this one represents both recessive traits. So because this wrinkled and green are both recessive, because round and yellow is dominant, then we know that this is 1/16, because you add this up, 9 plus 3 plus 3 plus 1 is 16. So 1/16 probability of getting a wrinkled green seed. And you could do the Punnett square out, and you'd see this square right here out of all 16. But it's going to take you a lot more time on the test than it would if you just memorize the ratios. So I'll show you how these ratios can help you. But I'm not going to use the Punnett square at all for answering the rest of these questions, because I don't need I know the ratios. And if you know the ratios, you can answer these too without doing a Punnett square. Okay, what's the odds of getting a round green seed? So round is dominant, green is recessive. I know that this is 1 dominant, 1 recessive. So I don't have to do the Punnett square. I know that the answer to this is 3/16. I know the answer to this is 1/16. I don't have to do the square. This one's a little trickier, and I'm going to tell you why. So we haven't really talked about this, but the probability of making this particular gamete is 1/4. And the probability of making this particular gamete is 1/4, because there's 4 of them. And remember, the rule of multiplication means that we can multiply 1/4 times 1/4, and that gives us 1/16. So the answer to this is 1/16. But you can also do it this way. So these are all, so let's just say, we'll do X and Y and Z, and we'll cross that to and Z. And so I might ask you, what's the probability that you're going to get this genotype out of that cross? Well, because these are each independent events, you can treat them as an independent cross. So if you do this cross, right, that's a monohybrid cross. We know the ratio is 1:2:1. So this is the one, so this is 1/4, right? I already know that. This I can also treat as independent. So it's a monohybrid cross and the ratios are valid. This, the odds of getting one dominant and one recessive are actually 2/4, which is 1/2, right? Because it's a 1 to 2 to 1 ratio. So this is the 2. And then again, this is also an independent event. So I can just do a monohybrid cross, use those ratios. The probability of getting 2 recessives is 1/4. And I can use the rule of multiplication. So this would be 1/4 times 2/4 would be 2/16. And then that times 1/4 would be 2/64 or 1/32. So the probability of me getting this would be 1/32. So I'll show you how we can do this with the question that we just asked. Okay, so we have big R, big Y, little y, and we're going to cross that to big R, big Y, little y. Same deal, right? So the probability of getting big R is from this monohybrid cross is 1/4. And and if we do this cross independently, the probability of getting big Y from that cross is also 1/4. And so remember, because we have the 1 to 2 to 1 genotypic ratio. And if you memorize that, you can do this super easy. Otherwise, you'll have to draw out a Punnett square. And then the rule of multiplication says I can multiply these together. So 1 times 1 is 1, and 4 times 4 is 16. So the odds of getting this are 1/16. And if you wanted to, you could go back and do the Punnett square on it, and you'd see the answer is right here. So only 1 out of 16 has this genotype. All right. So this type of cross, and I'll ask you on the test what this is because it's important. It's called a dihybrid. That means that it has one of each. It's a hybrid, which is the same as a heterozygote. And we're talking about di, so that's 2 traits. In this case, shape and color. But we could add another one, and then it would be a trihybrid. And you can also use math to figure out how many gametes it'll make. So if we're looking at one trait, there's 2 alleles, right? Because it's a diploid, and then the power is the number of traits that we're doing. So something that is diploid and we're looking at one trait like shape, then you would only make 2 gametes, right? In this case, it's either round or wrinkled. 2 gametes, right? And then we add, when we did 2 traits, which is this dihybrid cross, 2 to the second, so 2 times 2 is 4 gametes. And so, you know, without me redoing all this, I'll just go reference this other slide. But so we have 4 gametes now, right? Because it's a dihybrid cross. And if we were to do 3, so let's say we threw another one in here, and we'll just call it X. Then that would be a and this could be like plant height or something. I'm just making it up. So then we would have 2 to the third, which would be 2 times 2 times 2, which would be 8 gametes. So if you did a trihybrid cross, you would have 8 in a Punnett square. You'd have 8 across the top and 8 across the side, and that would be 64 squares. And if you were doing 4 traits, then that would be 2 times 2 times 2 times 2, which would give you 64 gametes. I think, no, 16. 16 gametes. So then you'd have a square that's 16 by 16, which I think is 256 squares. Yeah, and so on. So you can see how, if you're looking at an organism with 20,000 genes, your Punnett square would be trillions of squares in size, and it would take you forever to figure out the genotypes. So that's why it's super important to know the ratios and to know these little shortcuts. All right, so make sure that you guys learn those for the test, because it'll save you a ton of time and a ton of math, and you can check your work too. Okay, so that's in a Mendel's perfect world, right? We have complete dominance and complete recessive traits. There's no blending. Everything is independent from each other. It's not on the same chromosome, but that's in a perfect world. And when Mendel wrote this paper, nobody's sure if he got really lucky and all the traits that he looked at. So the ones we talked about here, all these different traits, all of these traits here, they just happen to be on different chromosomes. So we don't know if he cheated a little and he got some weird numbers and just threw those out, because by the time they discovered his paper, he was already dead. So he either got really lucky or he fudged his data a little bit, but for whatever reason, he never included genes that were on the same chromosome, and he never included any genes that blended together. But it does happen. So what we've talked about so far is like there's two traits. One is like a pigment, like purple, and the other one is default, like if there's no pigment, then it's white. And it would be kind of like moving into a house with white walls, and if you don't have any paint, you end up with white. And if you do make paint, then it's whatever color that paint is. So in this case, you could make a white pigment, right? They exist. I mean, you know that there's white paint. You could go buy it. Now, if there's really a white pigment and it's not a default, and there's really like a red pigment, which happens in snapdragons, then you would have a red paint. You could have a red paint and a white paint, and when you mix those paints together, what would you expect? Pink, right? So this has red and this has white alleles, right? It's really making a white pigment. It's not a default. This is really making a red pigment. So it's like mixing white paint with red paint, and in the end you get pink. So we call this incomplete dominance. Incomplete dominance means that the phenotype is halfway between the phenotype of the parents, the parental generation. Complete dominance is what we just talked about. So dominant phenotype is expressed in the heterozygote. In other words, if we were doing purple, then this would be purple, and it would be the same purple color as the parents, not a mixture of purple or a light purple. All right, so if we do this, then this pink flower has one red allele and one white allele, and it can pass on one of each, right? So either red or white, right, when it makes gametes. So it could pass on white, it could pass on red, and then the father of this would have the same genotype sperm. And so if this sperm fertilized this egg, we would get two reds, and that flower would be red. If this sperm fertilized this egg, we would get red and white, and so we would get pink. If this sperm fertilized this egg, right, we would get white mixed with red, and that will give us pink. And if this sperm fertilized this egg, we would get two whites, which would give us white paint. So the ratio here is different. If it's complete dominance, it's a 3-to-1 ratio. We'd learn that from Mendel. But if it's incomplete dominance, we would expect a 1 to 2-to-1 ratio, where this would be the dominant trait. It's kind of, there's not really a dominant, so it's in, but we just kind of name it. And then this is recessive, and then this would be halfway in between. So in this case, this is pink, this is red, and this is white. You could flip red and white around, it wouldn't matter because it would still be the same 1/4 ratio. So on the test, I might ask you, you cross 2 guinea pigs together, one's black and one's white, and you get 2 brown, black, and a white, and this happens with labs too, then is that incomplete dominance or complete dominance? And the answer is it's incomplete dominance, because if it was complete dominance, if I crossed a black and a white guinea pig, I would expect all the offspring to be black in the first generation. All right, so so these are terms that you're gonna need to know, and you need to know the ratio so you can identify what's going on. That's kind of the purpose of genetics. So there's a thing called codominance, and codominance means that both of the alleles are expressed in the phenotype of the offspring. So a good example of this is blood groups. So you probably are aware of this. So in a cell, you have 2 alleles for your blood type, and one you got from mom and one you got from dad. So you could be A, right? And you could have A on both, one you got from mom and one you got from dad. I'm gonna switch to red. And so that's gonna make a protein. It makes a protein that is actually a surface receptor. So this is an A surface receptor. And because there's only A's, the only thing that's going to be made on the surface of the cell are going to be A receptors. So just for shorthand, the sort of goalpost is going to equal A allele. And then I'm going to make like a V-shaped one for a B allele. All right, so this is what you would get if you had 2 A's. Now you could also inherit a broken A, so it doesn't make anything. We call that O. So you would make half the number. Let's say with 2 A's, you'd make 6. Right? So if you had, if you're AO, you would only make 3 of these receptors, because you only have 1 allele. But it doesn't matter because you're only presenting A. So in this instance, you have 2 A's. In this instance, you have an A and an O. Your blood type is A because you're presenting A on the surface of your red blood cells. Your body makes antibodies to things that are foreign. So the antibodies that you make are going to be anti-B. And I'll explain that in a second. All right, so the other possibility is you could be B blood type, and it's the same sort of deal. So you could have 2 B's, and that would make a cell surface receptor. Again, we can pretend like each B makes 3. So I'll just draw 6 out here. And so that would be BB, which was represented here. And BB means that you have B blood type. And AA means you have A blood type. You could also be AO, it's the same. And you can be BO, which is also the same, because this allele is broken. It doesn't make the right shape, so you're going to lose half of your receptors, but you're still presenting B, right? You still have B, and your body's going to make anti-A, because that would be foreign, because B is in your body, right? It's not foreign. This makes anti-B because A's are in your body and B's would be foreign. All right, so this is B type, and you could be BB or BO, and this is A type, and you could be AA or AO, and then this is anti-B if you're A type, and you make anti-A if you're B type. All right, so there's another possibility. You could have both an A and a B allele. Like you got one, let's say you got A from dad and B from mom. And then you make both of these receptors on the cell surface. So you would have both A and B on the cell surface, and so you wouldn't make any antibodies, because they would, if you made antibodies, they would attack A or B, and this would cause your cells to coagulate, and your blood wouldn't flow, and you would die because they all bind together. So if you have AB, you have one A allele and one B allele, and you make both surface receptors, and you're not going to make any antibodies, because if you did, it would attack yourself, and that wouldn't be cool. All right, and then the last possibility is that both of your receptors are broken, and we would call you O, because that represents the broken form of the allele. So nothing would be made on the cell surface. So in this case, you would be O. The only way to be O is to have both O's. That's what this is representing. And in this case, you would make A antibodies, and you would also make B antibodies, because A and B would be foreign to you since you don't have any A or B on your cell. So you would make both antibodies for A and antibodies for B. All right, so that's an example of codominance, right? If you have A and B, they're both made. If you have O and A, then they're both made, except O is the absence of it. A lot of students have a problem between codominance and incomplete dominance. And I guess the best way that I can describe this is codominance is expressing both, whereas incomplete dominance is expressing a mixture of the two. And it's kind of a gray area. So, but that's the definition. Also, blood type has to do with multiple alleles, because now we're not just talking about, are you purple or white? Are you red or white? Are you round or wrinkled? Are you green or yellow? Now we're talking about 3 different possibilities, like an A, B, and O. And that's sort of similar to like hair color, like you can have black and brown and red. And that's what we mean by multiple alleles, like it's more than 2. Anybody have any questions about this so far? Generally, when I'm teaching in person, students ask about, okay, well, that explains blood typing. And if you put, let's say that you put, if you're A and B, you don't make any antibodies. So you're safe to take anybody's blood. You can take all blood types. So if you're type AB and you're walking down the street and you get hit by a car, you can just grab a complete stranger and ask for a blood transfusion, because you would not have to worry about your body attacking anyone else's cells, because you don't make any antibodies to attack them. And O is you can only take O, because you make antibodies against A and B. So if someone put A blood in you, your cells would attack it. If someone put B blood in you, your cells would attack it. If they put AB blood in you, your cells would attack it. So the only type of blood that you could take is O, if you're O. But it's different because O has nothing on the surface, then there's nothing that the antibodies can recognize to attack. And so O is what we could call a universal donor. Because if you were O and you saw someone that was hit by a car, you could just offer to give your blood, because you know that it wouldn't be an issue. since there's nothing on the surface for the antibodies to attack. The AB, you can only donate to other ABs because you have both A and B on your surface of your cell, and AB is the only one that doesn't make antibodies. If you donated your blood to a B person, it would attack your A's, right? Because you have both A and B. And if you donated to an A person, it would attack, did I say that? If you donated to an A person, it would attack B. If you donated to a B, it would attack A. And if you donated to an O, it would attack both because O makes both antibodies. All right, so that's sort of a rundown of how blood types work. And then the question I get is, okay, well, what about the plus and the minus? So plus and minus is a different set of alleles, a different gene. It's called the RH factor. It was RH stands for rhesus. It was found in rhesus monkeys. And either you have it, which is plus, or you don't. So it's the same sort of deal. Let's say that you have RH factor here, it's plus, you're going to make a receptor for RH. And you could have a plus on this one, and you'd make double the amount of receptors, right? So you could imagine that you wouldn't want to make antibodies if you're RH plus, because it would attack your own cells. And then you could be RH plus and RH minus. You would still make RH, so you'd still be RH plus, but you'd only make half as many, kind of like the AA or AO. So this is still an RH plus, even though you're plus minus. This is an RH plus because you're plus plus. And then the last one would be RH negative, which means that both of these are broken and you don't get anything on the cell surface. It's kind of like O, where there's no receptors on the cell surface, so you make all the antibodies. And this can become an issue, especially like if you have a child that has a different RH factor than your own, because the antibodies would want to attack either the mother or the offspring's cells, and so it's really important to monitor that, because the antibodies, if they cross the placental wall, then it could be an issue. All right, but you don't need to know that because I didn't cover that in the lecture. I'm just sort of telling you that. All right. Some other genetic terms that are different that you need to know, so make a flashcard or whatever, however you can remember this. Pleiotropy means that you have a single gene that can have multiple phenotypic effects. So an example that your book talks about is sickle cell anemia. Normal cells carry oxygen to your blood, and so it's not an issue. Sickle cell, and we talked about this as a mutation. We talked about this in chapter 5. It's a mutation of a single amino acid in hemoglobin, and it causes the cells to collapse on themselves, and they form this sickle shape. Well, that sickle shape can't carry oxygen very well. So that causes you to be anemic, right? Because the red blood cells aren't formed properly. And if you don't get a lot of oxygen, you'd be physically weak. So that would be another phenotype. If your heart doesn't get enough oxygen, it can't make enough ATP, then the muscles it could cause your heart to fail. The sickle cells can also, because they're sickled, blood cells are round because they roll nicely through blood arteries. But imagine if you had a bunch of sickle shapes in there, they'd hook into your artery. This is supposed to be an artery. And so these could cause pain, because it probably doesn't feel good to have a bunch of things stabbing into your arteries, these sickle cells. It can cause blood clots because these all bunch up together, which could cause you to have a stroke, right? So it could cause brain damage. It could damage your other organs that filter your blood, like your liver and your spleen. And then this could lead to other phenotypic effects like impaired mental function, pneumonia, rheumatism, kidney failure, paralysis, and so on. So one single gene, one mutation in hemoglobin, one of the subunits of hemoglobin, can lead to all these different phenotypic effects. So that's an example of pleiotropy. A single gene has multiple phenotypic effects. All right. Epistasis. So epistasis is where you have two genes working together in a pathway. This is common where and we kind of talked about this like Roundup. There's multiple genes that is needed to convert sugar into amino acids in plants. And so these genes are in a pathway, kind of like you can think of it as an assembly line. So in this case, the example here is for mice color, but it also is valid for like dogs, like labs, where they can come in black or brown or white. And so what happens is that you have one gene, it makes the paint, the pigment. And once the pigment's made, then you also have another gene that puts the pigment in the fur, right? So think about it this way. You can think about this as the paint and this as the painter. So let's say you went and you bought some paint and you threw it on the floor. Is it going to get on your walls by itself? No. You need a painter to put the paint on the wall. So even though you have the paint, it does, it's not going to change the color of your walls unless you have a painter. So here, the gene for the paint, the pigment, is B. And so B is dominant. And little b is a brown paint. And in this case, the default color is white, just like the walls, right? So if you bought black paint and you had a painter, your walls would be black. If you bought brown paint and had a painter, your walls would be brown. If you had 2 gallons, one was black and one was brown, and you painted them together, the black would overpower the brown and it would look black. So that's what's going on here. So these mice have one allele for black, one for brown, which is represented here, and one for C is the painter. So if you have a big C, you have a painter. Little c is no painter. And we're going to cross that to another mouse with the exact same thing. So what is this cross called? Surprise question. It's dihybrid. It has both traits and 2 different genes. All right. So in a normal dihybrid cross, we would know that the phenotypic ratio would be 9:3 to 3:1. But in epistasis, it's not. And here's why. So we can make all of the things, and this is exactly like the P thing. So this one could give big B, big C, right? It could be big B, little c, which could be a gamete. It could be little b, big C, or it could be little b, little c. And so all of these are represented. And remember, we talked about this. It's 2 to the second. So that's 2 times 2, which is 4. So we know we did it right if we come up with 4 gametes. Sperm, this is the male, I guess. And this would be the female. Sperm meets an egg. All right, so I'm not going to go through this whole thing, but let's just assume this is right. The artist drew this right. This is going to have big B, big B. Well, what color is that pigment? It's going to make 2 coats of black right? And this means that it has two painters. So we have black paint and we have a painter. So what color is the mouse?
Black.
Yeah. And this one, we have one coat of black. That's okay. And then two painters. So we're going to make black and we have a painter. Then what color is the mouse?
Black.
And this is the exact same scenario. Now this one's weird, right? Because we don't have any black. We're both brown. So what color is the paint?
Brown.
And we have painters. So what color is the mouse?
Brown.
Right. And you could go through this whole thing and see. Well, we have two black cans of paint. We have one painter, but that's enough, right? It may take longer, but it doesn't matter. The wall's going to get painted. The mouse is going to get painted. So this one will be black. This one, black is dominant to brown. We have one painter. It's black. This is the same as that. So is this and this. And then we get to these. These are unusual. So this makes black paint. But notice we have no painters. So what color is the mouse?
White.
White by default. And same thing here. We have black, no painters. Black, no painters. Brown, no painters. So it doesn't matter what color pigment we make because we have no painters, so the mice are going to be white. And this makes a weird ratio. So we get a 9 to 3 to 4 ratio in epistasis. What happens here is that the 9 to 3 to 3 to 1 ratio, this one collapses into 4 because they're all blended together because we have no painters. You could also, another epistasis ratio, and this is how scientists discovered genes in a pathway for like cancer and all kinds of stuff by doing these crosses and counting the offspring. So in non-epistasis, we get 9 to 3 to 3 to 1. And we already did this in a dihybrid cross, which is what we're talking about. In epistasis, we get 9 to 3 to 4 because these two come together and give us this ratio. So if we see a weird ratio, we know that those are genes in a path working together. These two genes have to be in a pathway, creating the phenotype that we see. And in some cases, these all collapse together. So we end up with a 9 to 7 ratio. And that also tells you that the genes are in a pathway. And the fancy name for genes in a pathway is epistasis. So that's how you know epistasis. So on the test, I might say, you crossed a black lab and a yellow lab, and you got a 9 to 3 to 4 ratio of black to brown to yellow labs. What is that example of? And you would tell me epistasis, because it's a 9 to 3 to 4 ratio or a 9 to 7 ratio. If it wasn't epistasis, then it would be the Mendel ratio of 9 to 3 to 3 to 1. And so, you know, this is how we discovered the different genes that, you know, are involved in cancers or you know, we learned that there's like 20 different genes involved in height and stuff like that by doing these simple crosses and looking at the ratios. All right. So another thing is that, and you probably know this, that, you know, when we looked at Mendel's world, it was like, okay, there's one gene that controls color of the flower, but that's not always the case. In humans, we know of at least 3 genes that control skin color. And so it's pretty simple, actually. If you have one of these genes, then you're really light. Well, you could actually have 0. So if you had 0, you'd be super light skinned. And, you know, 2 would be darker, a little darker. And then 3 would be, you know, medium dark, and 4. And you could have up to 6 of these. And if you had 6, you'd be really dark skinned. And it's because each of these genes produces the pigment melanin, which causes your skin to be dark. And there's a reason for that, because dark skin the dark pigment absorbs light like a black hole. And remember, light can damage your DNA. So if you're getting a lot of sunlight, you want your cells to be dark so that the pigment grabs the sunlight like a black hole instead of your DNA, because it would screw it up. So you have less mutation if you have darker skin, and with more sun exposure, because the dark pigment is sucking up all the harmful radiation from the sun. All right, so if you lived on the equator, you'd get a lot of sun, and you'd want dark skin. If you lived at the pole where it's only light out, 3 hours a day, then you don't need to waste the energy to make those pigments. So you would lose those genes. It wouldn't hurt you to not have dark skin. And by default, the default pathway would be no melanin, which would make you have white skin. All right, so anyway, so you could have two people that are medium skin tone, right? And this is a trihybrid cross. And so the question would be, okay, what's the probability that you would have an offspring that had absolutely no pigment? And we could do these because they're independent events of each other. And so this is an independent event. Well, if this is a monohybrid cross, what's the probability to have this offspring? And remember, it's a 1 to 2 to 1 ratio. So this would be 1/4. This also the same deal. This is also 1/4. And again, 1/4. That this cross would produce this offspring. And the rule of multiplication allows us to multiply these together. So the odds of you having a completely light-skinned child is 1/64. And on the other spectrum, what's the probability of you having a really dark-skinned child? Well, it's the same. We just independently look at what's the probability of this, and it would be 1/4. This one also would be 1/4 from the big B, little b, big B, little b, and this one would also be 1/4. So 1/64th probability that you would have a really dark-skinned child. And you could do all these other ones in between, like let's just do this mathematically to see if it works out. So this one would be big A, little b, little c, little c. This offspring here. What's the probability of this? Well, what's the probability of getting a big A, little a from this cross right here? And the answer is 1/2, because it's a 1 to 2 to 1 ratio. So this is 2/4, which I reduced to 1/2. This is 1/4. This is also 1/4. So we can multiply these together, and we would get 1 times 1 times 1 is 1, and then 4 times 4 is 16, times 2 is 32, right? But you could also be this. I'm sorry, little a, big A, little b, little b, little b, little c, and that would also be 1/32. And then you could have one big B, and that would be another 32nd. So we would have to add them together. So 1/32 for one big A plus, so for this or this would be 1/32. Or if you had one B, right? So the rest would be recessive, all smaller case. Did I do this math wrong? One-fourth times 1/4 is 1/16. Oh, I don't have the common denominator, so I have to do this. sorry, so this is 2/64, which now puts me with the same denominator. So we would have 1/64 plus, sorry, this would be 1/64. 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 And this one would also be 1/64. So we can use the rule of addition because each of these offspring only have one dark allele. Everything else is recessive, and then if we add them all together, we would get 6/64 by using the rule of multiplication and addition. And that's happened, and you can look it up. If you look up, well, I'll just show you. So I'm just looking up dark and light skinned twins. I know that there's quite a few examples of this. Okay. So here's some examples of that. All right, these are actually twins. This is from National Geographic. You can see that they're from the same parents, right? These are twins right here, same parents, twins, same parents. So you can have extremes like dark and light skinned. And so that kind of goes against this whole Maury Povich thing, which, you know, he does all the, who's the baby daddy. And the most common thing that all of the fathers say is that that baby doesn't look like me. But that doesn't have to, because genetics is random chance. And so even though 1/64 might seem like a small probability, it's not really that big. I mean, people win the lottery all the time. So it can't happen. So my son told me a joke yesterday. He said, the probability of you being killed by a squirrel is really low, but it's never zero. And that made me think, yeah, that's true. So back to genetics, we talked about pedigrees already. I had you guys do the pedigree for extra credit assignment. So remember the rules of the pedigree. Squares are males, circles are females. I guess I should use my pen instead of my mouse. If they're not filled in, that means that they don't have the trait. If they have a dot in there, that means they're a carrier. If it's filled in, that means that they have that phenotype. So it could be, if you know you're doing a pedigree on brown hair, then you would fill this in for people with brown hair. If you're doing a pedigree of blonde hair, then you'd fill this in for blonde hair. And then these would be carriers of that allele for blonde. The horizontal lines are matings, right? And the offspring are shown below that. And then shaded symbols, like we just said, it shows that that individual has that trait. Pedigrees are really important. I'm just going to tell you guys that. So you probably heard of the breast cancer gene, BRCA1. This is the gene that Angelina Jolie had, and she had that mastectomy because of it. It also increases your risk of ovarian cancer. This is a super important breast cancer gene. It increases your risk of breast cancer by about 50%. And it's because you're a step closer to all of those failures that you have to have in cancer that we talked about in chapter 12. Anyway, we already talked about this pedigree, but the reason that pedigrees are important is that the reason we know about this breast cancer gene isn't because some high-powered scientist discovered it. It's because a nurse realized that a bunch of the people in her family were developing breast cancer. And so she had paid attention in her 181 class and decided that she was going to go back and reconstruct the pedigree on her family. And then she did, and she noticed that there was a trend. And because she was paying close attention to the chapter in genetics, she was able to identify how this was passed on and the genetics of the breast cancer gene. So a pedigree is actually responsible for the discovery of the breast cancer gene. And if no one did a pedigree on it, we might still not know if that existed and what the probability that you might develop breast cancer by having that gene would be without this stats. So I'm not going to go over this pedigree. You've already seen this pedigree, the royal family. And we know that it's bad to do inbreeding. There are other pedigrees that we could do. And one of those is for widow's peak. You could do this on your family or whatever. So widow's peak, having this peak right here, is dominant to not having one, which is recessive. And so you could sort of do a pedigree on your family just by these observations. And we have free and attached earlobes. And you have a survey for the population genetics lab that looks at your little finger, if it's bent or straight, and your earlobes, if it's free or attached. So you know from this that having an attached earlobe, so you don't really have an earlobe, is recessive, and having a free earlobe is dominant. Now, if you have a free earlobe, you don't know if you're homozygous dominant or heterozygous. But if you do have an attached earlobe, you know that you are homozygous recessive. All right. So this there will be some questions on the final exam about this. I think this link is still valid. All right, so we can probably look this up using the Fugates. Yeah, here's the link. I don't know why that control link didn't work with that, but we can just share it with you real quick. It's not screen sharing this. Here it is. Okay, so this talks about the Fugates of Troublesome Creek. It's they changed the website, I think, from this, so I'll need to update that to genie.com/projects /Fugates of Troublesome Creek 37.819. So anyway, this talks about it. It's a condition, a skin condition that causes your skin to be blue. And so what I really wanted you to look at is this pedigree. So the assignment, part of the assignment is to tell me what the gene is. And so that the gene is methaglobinemia. That particular gene has to do with oxidation reduction reaction. And so what I wanted you to see is, remember that double lines where we show inbreedings. So we look at this pedigree, we have 1, 2, 3, 4, 5, 6, 7, 8, 9 inbreeding events in this family that help fix this gene into the population, and it really causes your skin to be blue. So you can read more about that. We're going to switch back to the PowerPoint. All right. So what I want you guys to do is do a little research on the internet on this condition. It has a molecular mechanism behind it. And like I said, it has to do with the way electrons flow to and from hemoglobin. So as a bonus, and it's, I guess anyone that watches this is going to see it. I used to do this in person, and so if you didn't show up to class and I didn't record the lecture, you wouldn't be able to answer this. But I'm going to ask you questions on the exam, on the final exam, and I'm going to tell you straight up, here's the questions I'm going to ask you. So if you know the answers to this, you're going to get 4 free points from it. So, and actually that'll be 3 points each. So you get 6 free points out of this. Name the enzyme that malfunctions in the process, and we just talked about that. And then tell me what the enzyme is normally responsible for doing. And you can also look up Wikipedia in this, and it'll talk about it as well. So I believe that this link is still valid. Let me just double check it. Yeah. So this will help answer all the questions for the final, so that you can get your free extra 6 points just for doing a little bit of internet research. All right, so that is the end of chapter 14 in genetics. Just a couple of announcements. I am going out of town on tomorrow. So I'm not sure if I'll have good internet access or not. So what I'm going to do is, instead of risking that for Thursday's lecture, I'm going to go ahead and record the lecture on Thursday. So I won't have a live lecture on Thursday. It'll just be a pre-recorded lecture that I'll put the link in Canvas, and then you guys can watch independently on YouTube. And then if I don't have good internet, then I will probably have to do office hours on my phone. I'm not sure how that's going to go, but we'll give it a shot, and then I'll keep you guys posted through announcements. And then I will be back Sunday. Do you have any questions about anything so far?
Nope.
Okay. So the other thing that I want to show you real quick is that if we go to Canvas, so we go to Canvas and you look at the home page. So we're at home, and you scroll down. So below the PowerPoints and the crosswords and the study guides, there's these genetics problem sets. So I want you guys to practice these problems. They're going to help you for a lot of the questions that you're going to have on the exam. So one of the things that I wanted to sort of address is this this several black guinea pigs. So I'm gonna rewrite this, and then we'll do this practice problem real quick, and then I'll end the lecture. So several black guinea pigs of the same genotype. So we know that they're black, and we know that they have the same genotype. So that means that they got to be big B, little b, or big B, big D. I'm just writing this down, and I'll share it in a second. Were mated and produced 29 black So we know that they made, in the F1, they was 29 black and 9 white. So what would you predict the genotypes of the parents to be? Okay, so let me switch screens real quick. Okay, so I have this written up here. 2 guinea pigs, they're both black. We don't know what their genotypes are, so we don't know if they're big B, little b, or big B, big B. So it could be this, or it could be this, or it could be this. We just don't know what the genotypes of the parents are. This is what we're trying to solve. We do know what the phenotype of the offspring are. And so we can do these crosses. Well, for this cross, we know that we should get a phenotypic ratio of 3 to 1, right? For this cross, the only alleles that the parents can give are big B. So all the guinea pigs are going to be black. There's not a possibility that they can be white. So we can eliminate this. And then this last one, we have the same thing. And if you're not sure about it, you can do the Punnett Square. Big B, little b, big B for this parent, that parent, this parent. And you can see if you do the cross out, in this case, they're all going to be black as well, phenotype. So we can eliminate this. So this is really the only outcome that would give us white ones. And we can look at, we would expect 3/4 to be black and 1/4 to be white. And so, you know, this is pretty close to that ratio. We're like 30 to 10 would be like the 3 to 1 ratio. And so, you know, there is a little bit of variation here, a little, you know, like flipping a coin. It's not, if you flipped it 10 times, it's not always going to come up 5 times heads and 5 times tails. But we can eliminate the other ones, and so we know for sure this is the genotype of the parents. So that's the answer to the genetics problems. And then real quick, I'm just going to show you that if we go back to home and scroll down, that the answers to all these genetics problems, I worked them all out as well. And so you can see this is how I did it. We can actually find the percentages, the part of the whole. And so we know that it's a 3 to 1 ratio and none of the other ones work. So the only outcome for the parents would be they have to both be heterozygous. And then I answered 2 and 3 and so on. So you should do that. These are similar to the questions that you're going to see on the exam. And so make sure that you do those practice problems. All right. Well, that's all I got. So like I said, no lecture on Thursday. It'll be pre-recorded. And then I'll be back Sunday night. So Monday, I'll have regular office hours. And like I said, on Wednesday and Thursday, I should be able to do my office hours on my phone, if not on my computer. But I just don't know if I have all the recording stuff that I need on my laptop to actually do this. So I'm going to pre-record the lecture for Thursday. All right, so that's the game plan.
All right, sounds good. Thank you.
All right, have a good one.
Bye.
Bye.